Hello, my name is Pamela Camejo, and I will present the results of an assessment of the fungal and bacterial microbiome inhabiting the rhizosphere of plants belonging to the Solanaceae family growing in the Atacama Desert, an extremely arid desert located here in the northern region of Chile. By studying the microbiome of these plants, we can identify members of the microbial community contributing to the adaptation and tolerance of plants to this extreme environment, which is characterized by its high aridity, lack of nutrients, high salinity, and temperature fluctuations. For this analysis, we used high-throughput amplicon sequencing to explore the composition and diversity of both fungal and bacterial communities of the rhizosphere. We collected rhizospheric samples of 5 different species of Solanaceae plants, which in this figure are indicated by circles colored based on the plant species. Samples were also collected at different sites of this section of the Atacama Desert called the Talabra-Lejía Transit. Each of these sites is characterized by different environmental conditions, such as pH, temperature, and water availability. We then extracted the DNA of the microbiome of each of these samples and amplified and sequenced 2 genetic markers: the 16S rRNA gene for bacteria identification and the ITS2 for fungi identification. We processed the resulting reads using a known pipeline, grouped them based on identity into operational taxonomic units or OTUs, and classified them based on public databases. We found that in the case of bacteria, the microbiome of these plants was mainly composed by actinobacteria, protobacteria, acetobacteria, and Bacteroidetes phyla. But in the case of fungi, most of the OTUs were actually unclassified. We also calculated alpha diversity indexes for each sample and the relationship between samples diversity and environmental parameters. We saw that in the case of bacteria and fungi, their diversity was positively correlated to aridity, high precipitation, nitrogen, and organic matter, and negatively correlated to electroconductivity, which is a proxy for salinity, temperature, and pH, and in the case of fungi, to the potassium concentration in the soil. By generating PCOA plots of these samples, we also proved that the sample's microbiome structure was actually diverging among plant species and sample sites at the transect. And finally, we used the abundance data of each OTU to construct a co-occurrence network and also to group OTUs based on trend of abundance into different modules, where each module has OTUs with similar trends. In this network, each node corresponds to an OTU, bacteria as circles and fungi as squares, and the edges represent the correlation between them. Nodes are colored based on the corresponding phylum level, and the node borders according to the module of OTUs. We found 4 different modules here, and we also saw that the most connected OTUs were members of the Actinomycetales, a bacterial phylum known as plant growth promoter. However, the most connected fungal OTUs were unclassified, indicating that the Atacama Desert is a prime environment to identify and discover species that might have a relevant role in plant stress tolerance. Thank you.